Hello, everyone. My name is Harry He, your host today. And thank you so much for joining us for the webinar, Unveiling the PROTAC Revolution, Advancing the Drug Discovery Through Targeted Protein Degradation. Protein technology and also PROTAC technology has rapidly become a novel therapeutic paradigm. Today, there are already 10 PROTAC drugs in phase 2 clinical trials or beyond. And notably, the star product, AVR471, is undergoing phase 3 clinical trials and has received FDA Fast Track designation. This breakthrough underscores the transformative potential of PROTACs as a drug modality. As a revolutionary drug discovery and development strategy, PROTACs treat diseases by inducing the degradation of target proteins, providing a promising approach to challenging undruggable targets. And today we're very excited to be joined by 3 experienced industry experts, Dr. David Taddei, and Dr. Jean-Marc Garnier, and Dr. Michael Roy. And they're going to share their valuable insights into the linkerology case studies in PROTAC development and structure-guided optimization strategies. And we're also going to give about 10-minute Q&A section at the end. So please feel free to ask your questions and type them in the Q&A chat box. And you don't actually have to wait till the end of this webinar typing questions. Just put in your questions as you hear the talk. So before we dive into the presentation, let me take some time to introduce you our esteemed speakers today. The first one, Dr. David Taddei. As part of our global team as the program manager, David is based in Montreal, Canada, and oversees a portfolio of North American and European clients. He also works along Dr. Garnier on the development of Viva's chemistry enabling platforms like the PROTAC libraries. David is a seasoned drug hunter from his cancer career at Signature Discovery, where he led multidisciplinary research, including the discovery of inobrib, a first-in-class oral dual promodomain inhibitor drug against multiple specific cancer types, and several kinase inhibitors as inhaled candidates for the treatment of COPD and asthma. Now, I'll give the floor to Dr. David Taddei, and please feel free to share your screen and begin your presentation. Thank you.
Thank you, Harry. So to just start, our first presentation is going to be centered on the linker component of the PROTAC modality. And its title is Not Just Another Linker, Viva's Approach to PROTAC Linkerology. For the next 10 minutes, I'm going to present some highlights from the literature and linker development, as well as our modular approach to the PROTAC linkers and the linkerology building blocks that we use for our client projects, as well as bespoke orders. If you're not used to working with Viva as a global CRO company, our PROTAC platform actually mimics most of our drug discovery platform based on a holistic approach. That is to say, we can pretty much deliver from gene all the way to CMC, CDMO for small molecule biologics, as well as modalities such as PROTAC. As far as the PROTAC platform is concerned, one really needs a world-leading protein production and structural biology group with strong insight into crystallography. And very recently, we invested into our cryo-MEM technology, which is really well suited to the PROTAC modality. But also, we can delve onto our mass spec derived and biophysical technology to really identify new hits for PROTAC discovery, as well as the most traditional computational chemistry, assays, and medicinal chemistry DMPK SAROs. But all of these are really tailored to PROTAC discovery, and this is what we're going to try and cover as far as the linker is concerned over the last few minutes. So as far as the track record is concerned, I won't really presenting all of this slide, but we established it first in 2018, and have since delivered well over 50 projects now, and close to another 50 free-for-service projects, delivered over thousands of compounds, and are co-inventors on client patents. Our large groups of seasoned PROTAC chemists can tackle any of the season synthesis and purification on grams and tens of grams synthesis. So the know-how, including the DMA aspects, is really a strong suit of the Viva team. Specifically, I'm going to look at our custom build library, which was built off the shelf and really much presents how we address the linker and the linkerology challenges. Harry has already mentioned about the PROTAC modality and how it works, and Michael will really delve into the biology. So this illustration really covers in terms of the PROTAC modality as an hetero bifunctional molecule with two ligands, one ligand for the target protein and one ligand for the E3 ligase, which are linked together by the linker. Using the same color scheme, the characteristics of the PROTAC linkers are essentially to link those two ligands, as I mentioned, but they effectively enable through various chemistries, conformational directions, and as well as functionalities. They are essential and central components of this modality. So we characterize them by their length, their hydrophobicity, their flexibility, as well as the functional group that enable the attachments of both ligands. So there is a high structural diversity as shown on these three PROTAC from the literature. They also impact on the production of the ternary complexes, the degradation activity, and the target selectivity, as well as finally on the physical, chemical, and the ADME properties of those PROTACs. If you conveniently lose this very recent review from Acta Pharmaceutical Sinica from this year, you can still see that for the last 22 years, the flexible linkers, i.e. the PEG and the alkyl-based, still feature in over 2/3 of the actual reported PROTACs, as well as the most recent discoveries in terms of the relatively rigid so-called linkers, which are based either on heterocycles, cyclohexanes, cycloalkylamines, as well as purely aromatic-based linkers, and the spirose sp3 enriched. These recent developments are going to be the ones we focus on because they impact the most the physical properties and other key oral properties of PROTACs. So to sum up really, as far as the impact of the linker is concerned, one can really delve into the physical chemical properties and affect really heavily via discrete chemical modifications, the solubility, the molecular weight, number of rotatable bonds, as well as more traditional Med Chem metrics such as lipophility, TPSA, and hydrogen bonds. In terms of the ADME, the plasma stability can be really affected, as well as cell permeability and metabolism. Based on those, the pharmacokinetics and oral bioavailability can still be improved. However, I will see with the case studies, one has to rely on still orally designed ligands as far as the E3 and the protein of interest is concerned. And finally, you can affect big impacts on efficacy and selectivity. This can be summarized in those reviews, and those are going to be accessed through the recording at the end of the seminar. So based on what I just said, we still offer the so-called proof of concept kits, which are based on a variety of common E3 ligase binders, which are known in the literature, such as VHL and cereblon mostly, but also CRP and MDM2. And we are linking those to a variety of PEG linkers and carbon linkers. And the aim of this first pass is to really identify a minimum linker length, which is important to effective degradation. Secondly, you can, after developing these sets of 10 to 30 probes in parallel, your robust chemistry, you can also follow up with the linker composition and look at the so-called linkage and the direction, rigidification, the polarity of the linkers, working with this identified length range, as well as considering when to use negative control partial probes, which are available. Since then, I mentioned that there is a trend in the clinical candidates to go from the initial probes, which were, for instance, in the BRD4 and other targets, which looked very simple in terms of linear carbon and PEG linkers, And there is a clear trend as far as some of the disclosed clinical candidates to involve rigid sp3 complex linkers. So this kind of prompted us to add onto these proof of concept kits the linkerology kind of building blocks, which enable us to quickly assemble linkers in linker lengths from 2 to 20 atoms long, vary the end cap functionality and the attachment chemistry, as well as introduce some conformational rigidity. For instance, this illustrates how we can quickly, through diversity, increase the linker lengths. Secondly, we've got a range of bifunctional building blocks, such as those diamines, amino acids, and the likes, which are either mono or orthogonally bis protected to be able to quickly look at conformation as well as polarity and vectors conformational diversity in one pass. We also collaborate with other platforms such as the SYNthesis pyrochemistry platform, which was an internal platform used to generate pyrocyclic core on bulk quantities, which can be used in FT and as well as fee-for-service projects. And as you can see, those are mostly 4, 5, 4, 6, 5, 6 pyrocycles, and they introduce either a mono, bis, or tri kind of functionality for various vectorization. We also have partial probes assembled to various E3s, and on this slide, I will only introduce the second generation cereblon glutaramidide linkers, as well as the dihydrouracil, and they include some of the functionalities and bifunctional groups that I've just introduced. This selection of partial probes can also be bespokely generated based on the client requirements, and that can be a different E3 and different attachment chemistries. So as I try to summarize, our PROTAC in terms of linker design and linker accessibility is not based on sheer numbers. We try to assemble a modular and versatile chemistry-enabled platform based on spirocycles and other sp3 enriched building blocks, various sets of E3s, as well as dwelling onto some recent development to introduce some new and desirable ADME functional groups and building blocks, such as LED photochemistry, as well as using multi-component reactions and functional group interconversion to further enhance the versatility of the library. Lastly, I would like to introduce the VDEL TAC libraries from my colleague, Dr. Bing Jia, the VP of Early Discovery Platforms. So if you've got any interest or would like to know more about how VDEL and DEL libraries can be applied to PROTAC discoveries, please reach out to him and we'll organize, or even better, you can request his webinar, which was hosted a month ago. Finally, we are very active in terms of our recent developments to the PROTAC offering on LinkedIn. So if you want to follow us, please do so and reach out to our BD colleagues, Robin Wiggs, if you want to know more about our products. And that concludes this first presentation.
All right, thank you so much, Dr. David Taddei. Next, I'd like to introduce Dr. Jean-Marc Garnier. Dr. Jean-Marc is an experienced program manager in drug discovery, and he's led the medicinal chemistry of a first-in-class necroptosis inhibitor program, and has been named as an inventor in over 10 granted patents. With a PhD in organic chemistry, he has worked in various roles at Pfizer SRG, Peakdale Chemistry Services, and the Walter and Eliza Hall Institute of Medical Research. Jean-Marc joined SYNthesis in 2016 and is highly skilled in target validation, fragment-based drug discovery, and preclinical candidate selection. Now I give the floor to Dr. Jean-Marc Garnier, and please feel free to share your screen and begin your presentation. Thank you.
Thank you, Harry, and thank you everyone for being here. Hopefully you can see my screen now. Yes. Okay, so thank you for being here today. So yeah, So let me put my, so yeah, today I'm going to present a case study that we run or we are running internally at SYNthesis Med Chem on Viva Biotech. And during this case study, we will explore the strategy we use to transform or to repurpose a known POI binder into a new potent degraders. One of the questions we've been asked is why, or what is the reason to run this internal case study? And what we wanted to do here is to highlight how we could accelerate the discovery of new degraders and accelerate it through the design or through rational design with the use of a PROTAC acceleration kit that just David mentioned, and with the use of linkerology as well. And I like to do an analogy with solving a Rubik's Cube. So despite the number of moves possible or the complexity to solve a cube, if you run the right algorithm at the right time and in the right order, you can solve a cube in just a few seconds for the best. And this is kind of similar in our approach here. If we imagine we got all the pieces are mixed up or they're all scrambled, like some E3 ligase binders, some POI binders, or some multiple linkers. So yeah, all the pieces are scrambled, but we have all the information in hand. And the way we use this information and knowing how to use this information, when to use the information in one of the key to rapidly identify new degraders. And this is what we are going to discuss in this case study today, where we started from a known and patented BRD4 binder that has never been used previously for a degradation approach. We use some available extra structures for the design. We look at the best linker position attachment site. We look at the best linker position at attachment site. Then we look at the linker length and the linker rigidity with the aim of obtaining potent degrader. Our main focus. Sorry, I've got a problem. Our main focus was in obtaining potent degraders, but we also wanted to show that we could improve the in vitro metabolism of our degrader with the use of more advanced linker or advanced linkerology. So where do we start? So our starting point was to identify a suitable BRD4 binder. And we as I said previously, we wanted a POI binder that has never been used previously for a degrader approach. So we started with this 1-2-3 triazole BRD4 inhibitor series that had been patented and published a few years ago. And in the papers, so the author claims that there is around 30 analogs in the paper. The activity of the compound are between 3 to 50 nanomolar, tested on BRD4 by an alpha screen assay. On a couple of the best compounds were also tested in a cancer cell line with some further anti-proliferative effect with some EC50 reported between 100 to 1000 nanomolar. So the data here were really similar to GQ1. So GQ1 is a compound here. I'm not going to discuss GQ1 today because it's probably one of the most known tool compound, especially for the bed domain. But what I wanted to do here is just to highlight the key differences between one of the compounds from the paper called compound 20 on GQ1. So in terms of key differences, as I said, so this compound 20 has a 1-2-3 triazole versus GQ1, which has a 1-4 triazole. Compound 20 is a benzodiazepine, which means this is a 7-member ring with 2 nitrogen attached to a phenyl ring versus a thienodiazepine for GQ1. Both have a chloroaryl, but in compound 20, the chloroaryl is attached to a nitrogen, so a tertiary amine, and in GQ1, it is attached to a carbon of an imine. And one of the most obvious difference, if I can say this, is that GQ1 has a CH2 terbutyl ester, whereas compound 20 doesn't have any group at the alpha position of the triazole. But if we look at the binding site, the binding mode of these two compounds. So this is here an overlay of the 2 crystal structure of compound 20 on GQ1 in BRD2 domain 2. We can see they've got the exactly same binding mode, and they bind to the acetyl-lysine reconstitution pocket of the protein, making the same key interaction. So really quickly through the aspargine here via the triazole, or there is another interaction with the tyrosine via conserved water molecule, the same network of water molecule on the chloroaryl as well, filling a pocket called the WPF shelf pocket. So here, really what we got is compound 20 and GQ1 having the same binding mode. So we decided to use this compound as our binder for this case study. So the next step was to find the linker attachment site. And again here, so we decided to use some X-ray crystallography. So here we got a couple of ternary complex structure of two GQ1-based PROTACs. So the first one is a compound called DBET6. So this is a compound in green here. You can see it is in green here. So it is bound to BRD4 domain 1 on cerebrum. In this one, we can see that the linker is attached to the terbutyl ester of GQ1 via an amide bond. So we are thinking of doing the same in our case study. So attaching one of the linkers. So firstly, adding the CH2 terbutyl ester into the 1,2,3 triazole and attaching the linker at this position here. And I think it's worth noting that maybe I would say 95% plus of all the GQ1-based PROTAC compound have their linker attached at this position here. But there were also another compound, another interesting compound called DBET23. And for this compound, so the author attached their linkers through one of the, so this is a compound in yellow here, and the author attached their linkers through one of the thiophene of GQ1 via an amide bond. And we're thinking, as I showed you the overlay previously, we're thinking of doing the same on attaching our linker at the position that I call the position 5 here of the benzodiazepine. So these were our two strategies that we wanted to follow. So if we start with the strategy 1, so what we did is we started with this compound 20 that I just presented it. So we simplify it. So we remove this aminopyridine because we've shown that this doesn't really impact in terms of binding and doesn't make any key interaction to the protein. Then we added the CH2 terbutyl ester. And so maybe for the chemists in the room, you can see that we created an asymmetric carbon at this position here. And because we know that GQ1 has a positive, as a active and inactive enantiomer, so we decided to separate the two enantiomers here using some internal chiral SFC. So then once we separated the two isomers, we tested, or we look at the binding of these two isomers via SPR. So we had the SPR already set up in ours for BRD4 domain 1 and BRD4 domain 2. And just after testing the binding by SPR, we realized that there are very weak binding, especially relative to GQ1, with around 300-fold less activity than GQ1. But nevertheless, we wanted to still make a bifunctional compound based on from this binder here. So in order to do a direct comparison, so we use a compound already published in the literature. So this GQ1-based PROTAC here, and we use the exact same linker on the exact same E3 ligase here. So this one is reported with a DC50 of 62 nanomolar. So we prepare this compound called compound 2 here, and we tested it for its degradation, but we didn't observe any degradation with this compound here. So to try to rationalize this, so we did some modeling, and the modeling suggests that we saw some distortion of the diazepine ring, and we saw some clashes between the carbonyl of the terbutyl ester here with some residue of the ZL loop region. So this is probably why we lost a lot of binding for this compound into BRD4. So I think one of the key messages here is that the modification of the POI binder must maintain some sort of binding affinity to the POI. And it is known that we don't need a strong binder for a PROTAC approach, but you still need to make sure that the modification brought to the binder doesn't impact the binding too much. And I think it's always important to check it. So then we move to the second approach, the second strategy. So we did exactly the same. We started with this compound 20, and we remove this aminopyrimidine here, and we change it with a carboxylic acid with the aim of attaching our linker via this carboxylic acid or benzoic acid here. So then we tested again this compound by SPR on BRD4 domain 1 and BRD4 domain 2. And this time we confirm some binding of this compound to the protein. So then from this encouraging data, we decided to make our first generation proof of concept degrader. And in order to do this and to make sure that we could start to see some degradation, we use a really floppy or really flexible carbon linker here attached to a cerebrum binder or thalidomide. And so yeah, the first thing we did before test, so we tested the binding of this compound by SPR, and we found a KD of 570 nanomolar. And then, so we're ready to test this compound in our degradation assay. So for the degradation assay, we use an HI-bit luminescence assay. On this for a couple of reasons. So the first one is the assay was already developed and validated in-house, so it was really accessible for us. And the second reason is that the assay offers higher compound throughputs. We can also do a full dose titration, and it's more cost-effective and faster to run than Western blots. So yeah, so when we tested this compound, we saw a DC50, so degradation of 29 nanomolar with a full degradation as well, with a Dmax close to 100%. So that was really encouraging data. It shows that this exit vector 2 strategy was suitable to develop degrader from this scaffold. And then we were ready to go to the next stage, which was to use our acceleration kit. But just before I go to the acceleration kit, I just wanted to quickly come back to one of the importance of having a scalable advanced intermediate for derivatization. So this is something often overlooked, but important to avoid losing too much time in the synthesis process. So in our case, we found that the acid was not as easy to scale up as the pyrazole. So we decided to use the pyrazole as our derivatization compound. And we also observed a similar binding affinity between the pyrazole and the acid. And maybe another advantage of using the pyrazole is in terms of drug likeness. So we remove a potential amide bond early on, and this makes the pyrazole a more drug-like starting point. So now from this compound, we're ready to use our acceleration kit. And so in terms of acceleration kit, so this is as David mentioned really clearly just before me, so this is a pre-assembled kit of E3 binder with different alkyl or PEG linkers. So for this case study, we decided to use just a really simple E3 binder, so a thalidomide. And in terms of linkers, we also decided to use just some alkyl because we wanted to find the optimal shortest linker possible, just to avoid adding too much molecular weight to our compound, and also decreasing or not decreasing too much the log D of the compound. So I think this is really project specific. On every project we need a different acceleration kit, so this is really to be discussed depending on the different project, but in this case this is what we use, so just a thalidomide with some alkyl linkers. Here I'm just going to present some positive SAR just for the lack of time. I won't have time to go through the negative SAR. And just very quickly, so as I said, we use the thalidomide, but we didn't find any big difference in terms of degradation between the 4 or the 5 position of the thalidomide. And similarly with the heteroatom exit vector, so we try the oxygen and nitrogen without so much differences between the two. In terms of linker length, so we found that having a 4 carbon or 5 carbon was really the optimal linker length that gives the best degradation. And as I just mentioned previously, so we use the pyrazole rather than the carboxylic acid as an attachment point. I also added some representative example of SPR sensogram and HI-bit luminescence data, just to show that some really great binding by SPR observed for some of these bifunctional compound, and also a really nice dose response for this compound in terms of degradation with a full degradation observed. So in terms of the data, the KD, as you can see, they are really low KD, so between 10 to 20, 25 nanomolar in BRD4 domain 2. So for the base compound, so with 4 carbon or 5 carbon on an oxygen linker at the 4 or 5 position of the thalidomide. And in terms of the degradation DC50, we also find some a single digit nanomolar degradation with a full degradation for all of the compound. So I think the key message here is really that by using an acceleration kit, we can really very rapidly identify potent degrader with the optimal or the shortest linker length. So that was, again, really encouraging data. We had some really great compound in terms of potency. So the next stage was to check all the stability or the ADME property, and we really focused on the macrosomal stability. And as you can see here, the first compound tested were not great, and we saw some very high clearance observed. So we tested them in a mouse liver microsome, and some very short half-life, as well, less than a minute. So this is why we decided to do a bit of linkerology to try to improve this macrosomal data. And so this is what I'm going to present in the next couple of slides. So the first example I'm going to show you 2 examples. And the first example I want to show you is around using some advanced linker and using some rigidification or cyclization to improve the metabolism of the compound. And so we started from the C4, alkyl that I just presented previously. So the first things we did was to cyclize the C4 and make this cyclobutyl amine here. So what we wanted to see is really that if we start to rigidify our linker, do we lose any binding or any potency or any degradation potency of our compound? So what we can see from this data here, we don't really lose much, so maybe in the 2 to 3-fold only, but we still have a very strong degrader in the 7 nanomolar range with a full degradation. By doing this, we didn't really improve the metabolism of our compound. So this is why we decided to further rigidify this linker by doing some conformational restriction. And in order to do this, so we wanted to keep the similar length of our linker. We didn't want to increase the linker length. So we decided to swap this hemides here first, just to be able to do a further cyclization. So we tested this compound here just to make sure that the data were comparable between the reverse hemides, so which it was, so same DC50 activity, and also I think importantly to check that by doing this it doesn't change the macrosomal stability of the compound. So when we had this, we are ready to internal cyclization, and this is when we made this piperidine compound, so this piperidine amide with a cyclobutylamine, and this is where we saw some remarkable improvement in terms of metabolic stability. So we go from a 10 minutes half-life in mouse to a more than 300 minutes half-life in mouse with a very low clearance now. And it's also probably worth noting that we improve the DC50 by around 1 log unit by doing this. So I think really what I wanted to show here in this slide is the profound effect of the linkers. And as David mentioned, it's not just a linker. So the linker can have a really, really profound effect in terms of metabolism. So we started from a compound with a half-life of less than 5 minutes and going to a compound with a half-life of 305 minutes. So the linker is definitely not just a spacer between two binders. And the second example I wanted to show is we can use even more uncommon linker or more advanced linkerology, and still manage to improve our metabolic stability. So what we did here, so this is a compound I just showed you in the last slide, and we replaced this cyclobutyl amino acid and use this 1,3-dioxynyl or dioxynyl compound. Yeah. And by doing this, we can see we even further improve the stability of the compound, and now having a compound really, really stable with some half-life greater than 500 minutes and very, very low clearance for this compound. So what I really wanted to show here is there is still room, to find some even uncommon linker, and I believe no one never seen this kind of linker in the literature previously. Again, my key message here is the importance of the linker in the degradation and in the ADME properties. And linker with the right linker, we can really play around the macrosomal stability of the compound. Just worth noting as well that we are doing some further exploration at the moment. We tested one compound in an in a mouse in vivo PK to get his profile, and we saw a really good correlation between the in vitro and the in vivo clearance, so which means that we now have some compounds suitable for some in vivo efficacy model. And I'd like just to finish out doing a summary by doing this snapshot here and saying, how we started from a known compound, so a published inhibitor, and transform this one to a binder, finding the right attachment site to this binder. So and then from this one, making a first proof of concept compound. Then when we know that we've got a binder attached to a linker on cerebrum and can degrade the protein of interest, we can use an acceleration kit and just quickly identify the right linker length, the shortest linker length, but the right linker length. Having a really potent degraders, but lacking some macrosomal data, and then using some linkerology to really improve the ADME property of the compound, and obtaining some really potent compound and with some really good macrosome stability compound. So really, it's really it looks simple like this. So linker attachment points, using an acceleration kit, and then doing some linkerology to go from a binder, a non-binder never used previously, to an advanced degrader. And with all of this, I would like to thank all the persons that have been involved in this project. So firstly, thanking all the persons that work on the POI binder study, so from this paper and from this patent here, and also thanking all the persons working the degrader case study, so all my SYNthesis Med Chem colleagues, all the synthetic chemists, the medicinal chemists, but also a big thank you to all the PROTAC catalog teams that work hard to work on this catalog that we can offer. And I would also like to thank all my Viva colleagues, so the bioassay teams that tested the components, the HI-bit luminescence assay, all the SPR team, all the protein production team as well, the Viva protein production team that provides the protein for the SPR. They provided some protein for the cryo-EM and also provided some antibody for the bioassay for some of the Western blots. The DMPK team, all the CADD team as well for the modeling, and all the cryo-EM team as we are trying at the moment to get a structure of a ternary complex of one of our compounds. And with this, I would like to thank everyone for listening today.
Okay, thank you so much, Dr. Garnier, for your wonderful presentation. So today we also have the honor of hearing from Dr. Michael Koy. Dr. Koy Roy is a leading medicinal chemist, structural biology, and is currently based in WEHI in Melbourne. Dr. Roy, his research centers around targeted protein degradation, utilizing small molecules, chemical biology, and structural approaches to investigate and target cancer signaling pathways. During his postdoctoral work at the University of Dundee, under Professor Alessio Ciulli, Michael played a significant role in developing ACBI1 and ACBI2, which are degraders targeting SMARCA2/4 subunits of the BAF chromatin remodeling complex. Recently, as a project director in an international PROTAC collaboration between WEHI and Boehringer Ingelheim, Michael has co-led a team of talented researchers focusing on developing oncology-related PROTACs. All right, now I'll give the floor to Dr. Michael Roy. Please feel free to share your screen and begin your presentation now. Thank you.
Thank you very much. It's a great pleasure to be able to present today. So just a moment. Okay, so hopefully you can see my screen now. So today I'll get right into it. I'll be talking about some of the work being done to and ways that you can leverage structure and biophysics in rational PROTAC optimization. So this is work. I'll give two short case studies. So they are both work done when I was in the lab of Alessio Ciulli. I'm working with Boehringer Ingelheim in Dundee. But just to acknowledge also that I've recently been leading a WEHI Boehringer collaboration with really talented researchers. So we hopefully be able to talk about some of that work soon. So as has been alluded, a PROTAC needs to do a number of things to enact profound degradation, but one of the key steps, at least transiently, is formation of a ternary complex that's productive to enable ubiquitination on an accessible lysine. And I like to think, and I know many others think of molecular glues and PROTACs as sort of in many ways a spectrum where you have bivalent PROTACs that have independent binding to the E3 and the target. On one end, molecular glues remodel the E3 surface to enable the target binding. But in between that, you can really take steps to sort of glue PROTAC interfaces through careful linker re-engineering, and I'll talk about that today. And one of the useful measures that we can have for how we can identify stabilizing and destabilizing interactions in PROTAC complexes is using this parameter known as cooperativity. And so you can define that by measuring the binary complex of the PROTAC for the E3 ligase, and then the ternary complex, KD, where the PROTAC is already bound to a target protein, and then measuring for the E3 ligase, or you obviously can switch those around. And the ratio of these parameters, if you have a positively cooperative complex, you get additional stabilizing interactions and a positive value. So I'll just give an example of how we use that in developing potent degraders of subunits of the BAF complex. So the BAF complex is a chromatin remodeling complex that's mutated in about 20% of human cancers. So it's quite a profound cancer target. It's a challenging target because what we're actually targeting was this ATPase subunit SMARCA2 and SMARCA4. These are mutually exclusive subunits of the BAF complex. And what was known was that if you knock out SMARCA2 or SMARCA4, that has implications for potentially treating AML, but also many cancers have mutations in SMARCA4, and there's a synthetic lethal interaction with SMARCA2. This protein is large, or these proteins are large. They have a helicase domain, which at the time was deemed quite challenging for drugs, but there were ligands already available for the bromo domain. But unfortunately, they didn't phenocopy the effect of a genetic knockout. So we were interested in seeing if we could repurpose these to develop degraders. So we started from this SMARCA2 bromo domain ligand that had reasonable binding for the SMARCA2 and SMARCA4 bromo domains, identified a suitable exit vector from a crystal structure that had been solved at Boehringer Ingelheim, and this is one of the early compounds of a limited set that were made for this at the time, using a VHL binder linked through a relatively flexible PEG linker, and this compound showed only partial degradation of both SMARCA2 and SMARCA4 bromo domains. With a DC50 of about 300 nanomolar. It was recognized that this compound had relatively limited permeability, and also we could see that it lost quite a lot of binding for the SMARCA2 bromo domains by SPR. But in spite of this permeability and loss of binary binding, it still showed some degradation. So I was tasked with measuring the binding and cooperativity of this compound, and found that actually it bound relatively well still to VHL, and it had a positive cooperativity of about 5, which is modest, but it suggested that actually this was forming productive complexes because it could degrade, and this might be a way to try to optimize these compounds further. And this was really streamlined because early on, I was able to solve a structure of a ternary crystal structure of this PROTAC with VHL and the SMARCA2 bromo domain. And what was, and this was at the time one of few crystal structures available of this nature. And it showed it had, the flexible linker formed a close interaction with a lipophilic shelf of the protein, and we could really identify quickly a point where we could rigidify this linker to try to optimize this compound. So in really quick iteration, a few compounds were made where we could cyclize to rigidify that linker, this PROTAC2, and then extending this a little bit further with an ether in the linker, ACBI1, and this rapidly led to much more potent compounds. So what I've shown here is the binding to VHL, where we have in black is this initial PROTAC can displace a VHL tracer, but you have positive cooperativity and leftward shift of this curve when you have SMARCA2 bromo domain there. ACBI1, this compound can equally displace VHL, but we have a much greater shift when we have the SMARCA2 bromo domain indicating positive cooperativity. So from this crystal structure, we could also show that there was some really close interfaces between the bromo domain and VHL, and this arginine of VHL provided one of the key interfaces there. Mutating that arginine actually reduced the cooperativity significantly, supporting this crystal structure was relevant in solution. And so the net result of this was that we could very quickly optimize from a PROTAC with a flexible linker that had modest modest degradation to a PROTAC that had slightly improved binding to SMARCA2, but not significantly so. But really, the bigger difference was in actually the cooperativity. We could lock in a productive complex. Adding a little bit more lipophilicity to the linker also helped with passive permeability. And this really was borne out in a much more potent degrader in cells. So going from 300 nanomolar partial degrader to a degrader that had single-digit nanomolar degradation of SMARCA2, 4, and a closely related PBRM1 bromodomain with Dmaxes of greater than 90%. And this was reaching the DC50 within 2 hours at 1 micromolar. We also, I won't go into the biological testing in great detail, but we also did some proteomic studies to show the global proteomics to look at the off-targets and on-targets of this PROTAC. And you can see that the only proteins that were significantly degraded were in this experiment, SMARCA2, SMARCA4, and PBRM1, as we expect based on the selectivity profile. But interestingly, this is work done by Tom Owen Hughes' lab. When you immunoprecipitate the BAF complex itself, you can then see some loss of other components of the BAF complex, which seem to be particularly most likely those that are destabilized when you lose one of the core subunits, being the SMARCA2 and 4 ATPases. Okay, so in light of time, I'll move on to the next case study, which is quite a short one. And so this was really just to outline, obviously, a ternary complex is a really important intermediate within the degradation pathway, but can we actually measure using biophysics how long lived that ternary complex is, and whether that can inform on degrader understanding or optimization? So we used this in this example, MZ1, which is a really well-characterized VHL-based degrader of BRD4 developed in Alessio Ciulli's lab. And it recruits, amongst the BET bromodomain family, it recruits via the second bromodomain of BRD4 to form really cooperative ternary complexes with VHL. So to look at this, and this is now an approach that has been used by quite a number of labs, we immobilized VHL on the SPR chip, could measure really nice binding affinity for the MZ1, and then incubated the PROTAC with an excess of BRD4 BD2, and we could see a really profound extension of the off rate of the complex stability of this ternary complex, and this correlated also in a positive cooperativity. And then what was quite interesting was when we then related that to the selectivity, we tested with other bromodomains within the BET family and related this to the selectivity that we see in cells from kinetic experiments looking at degradation of BRD2, 3, or 4. We would always see that BRD4 was degraded very quickly with a really rapid initial rate of degradation and very profound. BRD3 had a much slower initial rate of degradation, and BRD2 also had a fast initial rate, but then tended to slow later on. With what was really apparent with BRD3, BD2 in this SPR assay was that you had much shorter lived ternary complexes. And we were fortunate, and we had a crystal structure of the ternary complex of MZ1, which when we overlay with BRD4, BD2, and when we overlaid that with the other bromodomains, you could see that there was one key amino acid that was a glycine in BRD2 and BRD4, but was a glutamate in BRD3. And that an overlay suggested that this would clash actually in this particular PROTAC ternary complex. So to see whether that was indeed the reason for this difference, we performed the swaps of this particular residue, and you can see based on the SPR traces that changing the BRD4 glycine to a glutamate, you get this profound drop in the lifetime of the ternary complex. and with a reverse swap, you extend the ternary complex. So this, I think, just really illustrates how this sort of SPR approach can be really valuable to understanding how a single amino acid can, for example, give really profound differences in rates of degradation and selectivity. But I hope I've also shown how structural and biophysical insights, in particular, if you can manage to get ternary complex data, can really leverage to rapidly optimize linkers, and you don't necessarily require cooperativity in all cases, but where you can leverage cooperativity, you can rescue often weaker binders to quickly enable fast, profound degraders. So with that, I'll hand over, and yes, I guess if there's any questions.
Okay, thank you so much for Dr. Roy for your wonderful presentation. So now everyone, now let's move on to the Q&A session to have a bit more interaction with our audience. And just a reminder, and if a question for a specific speaker, please type in the question with the speaker's name to direct the question to them. Let's see what question that we have. I already see some questions that already popped up in the Q&A section. Okay, all right, since this one is for Dr. Jean-Marc. So Dr. Jean-Marc, have you tested your best compounds in a solubility assay?
Yes, so short answer is yes. So we started to test some of our compounds. So just only a subset of compound in our solubility assay. So what we are trying to do at the moment, as has been shown in the literature, is testing PROTAC compound in a normal kinetic, I would say normal, kinetic solubility assay with PBS doesn't work well. On most of the time, the compounds are insoluble, so they are really difficult to classify like this. So it is well known that it's better to test the solubility in a thermodynamic assay using some biorelevant media, like some FASIF or like some FASIF media. But what we are trying to do is see if we can, instead of using a thermodynamic assay that use a lot of compounds. So usually for a thermodynamic assay, you need between 10 to 12 milligrams of compound, which is quite a lot, for when you are in heat to lead or early lead up, usually you don't make a lot of compound. So we are trying to see if we can use the kinetic solubility assay in a biorelevant media at the moment, and doing a comparison between the two. And what we did is we found that using the kinetic solubility assay in a biorelevant media, we are able to see some solubility of some of our compound, and we're able to classify the compound based on their solubility. So to be really honest, at the moment, the solubility is not great. So it's only, the best compound are around 30 micromolar in the solubility assay, in the kinetic solubility assay in a biorelevant media. So we still have some work to be done. Well, we still have some work to do here to improve the solubility of the compound. So there is, a lot of things that can be done to improve the solubility of our compound. So yeah, the first thing, of course, is to change the E3 ligase. So at the moment, we are just using I would say a plain E3 ligase. So there is some more drug-like E3 ligase, and a bit like David has mentioned previously. So there is a range of much more drug-like E3 ligase than can be used. And maybe the other point I wanted to mention here is when you start from a non-soluble E3 ligase and start from a very insoluble binder as well. So the linker can't rescue all the solubility. We've seen that the linker can rescue all the macrosome stability, but the linker can't rescue all the solubility. So we really need to work as well on the two other parts, so either the binder or the E3 binder, or the binder to the PY binder or the E3 binders. So yeah, this is what we are doing at the moment, trying to change this thalidomide binder to some more drug-like binder to see if we can improve the solubility of our compounds. So yeah, I hope he answers the question.
Okay, I think the next question is for Michael. So how do you view the advantages and limitations of the directed biology approach versus the SBDD approach in PROTAC discovery? And how do you foresee these two methods evolving and complementing each other in future PROTAC development?
Yeah, it's a great question. I think the directed biology approach, so whereby you do sort of parallel screen or synthesis and sort of crude screening of compounds in a biological system directly is very attractive and can really accelerate identification of hits in the first instance. And often, if you're lucky, you might get hits that have, if you have a diverse linker set, that you can have really interesting linker variants there as well. I don't think that, I mean, the two are highly complementary, obviously, and the examples I've shown where you can leverage actually a much smaller scale, sort of more traditional, I guess, route of PROTAC synthesis. But if you have access to great biophysics and structural biology, that can really accelerate, so that you don't necessarily need to make as many compounds to get to PROTAC to potent degraders. But I think that either way, if you're starting from a directed biology approach, typically these sorts of projects will then, as they move on, engage significant aspects of structural biology in terms of optimization. You can, as has been shown, even binary structures can really guide aspects of linker design and hypothesis generation. But I think where it's moving, cryo-EM obviously is playing a big part in helping in the structural side for PROTAC complexes with less protein requirements. I think also proteomics has its role in higher throughput analysis of ternary complexes, for example. But equally, you can one challenge, I suppose, of direct biology approaches is that if you're going straight to cells, you can obviously measure aspects of permeability and target engagement, but you can sometimes miss compounds that have really strong cooperativity effects or might be good degraders. So that's where I think building in, at least for selected compounds, some of the biophysical analysis is really valuable to see how you're tracking, and sometimes measuring those disconnects between degradation and compounds that might show relatively poorer permeability or binding, but still show profound degradation, can actually hone in on compounds that have properties that might be useful to optimize and go from. So I guess, do any of the other panelists have comments on that?
Okay, thank you, Michael. I see another question for Jean-Marc. So this audience said, the data presented is very impressive, and the development story and examples of improving potency and stability are very interesting. I was wondering if you could comment on the total number of compounds you made, not just the selected examples. Not needing to build the linker ligase is a huge head start, but does the acceleration kit provide efficiencies in this area, reducing the number of compounds made?
Yes, great question. Number of compounds, top of my head, we didn't make too many, to be honest. I think we made between 25 to 30 compound maximum in this project here. So yeah, acceleration kits, definitely improve or speed up the process. So this is what is called an acceleration kit, but because this is already everything is already pre-assembled, so you got already your E3 ligase linked to your linkers. So it's only one step to couple to your E3 binder. So this is a really powerful method to speed up the discovery of new binder. And I think what is important with this acceleration kit is you can really quickly find the optimal linker length as well. And then when you've got this optimal linker length, you can start to play around the linker to improve the property of the compound. So it is really an acceleration kit, and you can save a lot of time in terms of synthesis by using already pre-assigned E3 binder plus linkers. And as I quickly mentioned during the talk as well, so this is really project dependent. So every project would be different. Every project will need a different E3 ligase. We didn't talk about specificity or selectivity today, but it is well known that some of the ligases are not tissue specific or cell specific. So there is really really a lot that can be done here to accelerate some PROTAC projects. I don't know if someone else wants to comment further.
I think there is a risk as well, based on the results you've shown, Jean-Marc, today, is that you can really be bogged down by a number of things. Your two exit vectors showed a very different linker lengths. So those acceleration kits may definitely vary in terms of the compound numbers produced based on your linkage. So your pyrazole and acids were much shorter compared to the JQ1 amide direction. So if you were to tackle various linkages, you would have to take this into account as well in terms of selecting your lengths. And this is where you wouldn't just use the kits in isolation. You would have either some structural or some modeling elements to your programs. So I think the kind of numbers may be representative based on a number of kind of prior art disclosures, but the theme applies. We definitely will be able to speed up with a toolkit and a kind of combination of D2B type chemistry to generate libraries faster and more efficient.
Okay, I think we have time for one last question. So this question is for David. So how can we achieve specificity when you design the PROTACs?
Sorry, I couldn't get that, sorry.
So the question is, how can we achieve specificity when you design the PROTACs?
So, I mean, in terms of the linkers, there's been a number of reports. Some of the ones that are presented on the slides are one from the Sully lab, where forming actually a macrocycle, which, from property point of view, may be defying the kind of beyond the rule of 5, but nevertheless, the specificity was achieved and remarkable that way. And another one is actually more subtle, is discrete chemical modifications have been shown really to change the selectivity versus various proteins of interest. So, and just because you're starting to get some SAR with a certain linker lengths and functionalities doesn't mean that it's always going to translate across series, because every time you change the functional groups linkers, anchors, you will also affect the PPI interface. So this is going to impact not just the one protein of interest, but potentially others as well. So it's simply not a matter of identifying a vector and then going for an acceleration kit. You will always need to consider your counter screens as part of your optimization cascade.
And if I can just add to that, I think this is where I think structural knowledge can be really valuable. Often it can be just serendipity to identify sometimes more selective complexes, but also you can, as I've shown, you can really start to understand how particular residues and differences between highly conserved proteins you can engineer selectivity. And sometimes even that can bias design in terms of trying to push the complex in a particular direction with particular interfaces to bring them closer together to leverage the small sequence differences you have. But I think also, it's really clearly known in the field how global proteomics is really valuable to, looking in an unbiased fashion to validate what other off targets you might have that you didn't even realize. And I think that's really critical to just to know that simply because you have a selective binder, or in some cases a non-selective binder, the profile of a degrader can be vastly different to what the constituent binder might be.
Okay, all right. So I think that concludes our webinar today. And thank you all for joining us today. And also want to thank our speakers, Dr. David Taddei, Dr. Jean-Marc Garnier, and Dr. Michael Roy for making this event possible. And our dear audience, if you have any questions or feedback, please feel free to contact us. You can also check out our recording of this event by following our YouTube channel and also our official website. Again, thank you all for your time, and we look forward to seeing you next time. Thank you.
Thank you, Harry. Thanks, everyone.
Thank you.
Thanks, Harry. Thanks, everyone.
Thank you, everyone, for your interest.